Welcome. In this session, I'm going to start a new chapter, Molecular Basis of Inheritance. In the previous chapter, we understood how characters are passed from one generation to another generation. Now, in this chapter, we will understand the same inheritance at molecular level. So that's the chapter name, Molecular Basis of Inheritance. Right, so let's begin this session. So first we'll see about genetic material. So the question is, what is genetic material? So here genetic material is a one that stores information about all the characters of an organism, the skin color, hair color, texture, eye color, whatnot. So every character of an organism, its information is stored in the genetic material. So genetic material stores information, then it controls the expression of characters and is passed from one generation to another generation. So it passes from one generation to another generation, right? So that's what we call a genetic material. And we'll see now the properties of genetic material. So here, a molecule to be a genetic material, so what it has to follow. So properties of genetic material. So for a molecule to act as genetic material, so it should be able to replicate by itself. It should be able to replicate by itself. Now we know in living organisms there are various molecules, like of course fats, carbohydrates, and then proteins. None of these have a property of replicating by themselves, so hence they cannot act as genetic material. So for a molecule to act as a genetic material, it should be able to replicate by itself, and then it should be structurally and chemically stable. So the molecule should be structurally and chemically stable, and then it also should be allowing slow changes. So it should provide scope for slow changes, and these slow changes will actually help in evolution. So these are necessary for evolution. So that is a gradual transformation of a species into another species. Okay, so that's another point. Now it should be able to express, it should be able to express itself in the form of Mendelian characters, in the form of Mendelian characters. So it should show dominance, recessive features, right? So we understand that for a molecule to act like genetic material, it should follow all these. Okay, that is should be able to replicate by itself, should be structural and chemically stable, should provide scope for slow changes that are necessary for evolution, and should be able to express itself in the form of Mendelian characters. Then only, okay, that molecule will be a genetic material, right? Now let us see what molecule is acting as genetic material in living organisms, and we'll also see about non-living things like viruses and viroids. Okay, so we'll see viroids, then viruses. So these are non-living, and all the living organisms mostly, okay, they are included under 5 kingdoms. So they are included under 5 kingdoms. They are Monera, then Protista, Fungi, Plantae, and Animalia. So majority of organisms have been included under, okay, these 5 kingdoms, and all these will be including living organisms, right? And we know, so these 4 kingdoms are eukaryotes, and okay, so this one is prokaryotes. So it's the only kingdom, Monera, includes prokaryotes. The remaining all 4 kingdoms, they include, okay, eukaryotes. Now coming to the viroids, in this case the genetic material is RNA. The genetic material is RNA. And coming to viruses, in them the genetic material is either RNA or DNA, but not both. So I told you in previous sessions that no DNA will have both RNA and DNA. So because of that, we classified viruses as DNA viruses in whom the genetic material will be DNA. And in case of RNA viruses, RNA will be the genetic material, right? Now coming to the prokaryotes and eukaryotes in all living organisms. So they have both RNA and DNA, right? In case of bacteria, they have RNA and DNA. In case of protistants, they have RNA and DNA. In all living organisms, that includes prokaryotes and eukaryotes, all of them have both RNA and DNA. But the RNA will not act as genetic material. So here only DNA acts as genetic material, even though, right, all the living organisms are having both RNA and DNA, only DNA acts as genetic material. Now the question comes, what about the RNA, right? So here RNA acts as non-genetic material. So it is not, so it will be not the genetic material, so hence we call non-genetic material. So what will be the function? So RNA acts as adapter molecule. So what is this adapter molecule? How it is acting, we will understand only in our coming session. So at present I'm just giving you an introduction. Okay, so acts as adapter molecule, and it can be a structural component, like in case of ribosomes. So the ribosomes are made up of these RNA and proteins. Okay, so that's what the RNA will be acting as structural component in case of these living organisms, all the living organisms, right? And then it can also act as catalytic molecule, right? So some of these we will understand. So whatever I have mentioned here, so that RNA acting as adapter molecule, what we mean by that, okay, structural components, catalytic molecule, all that we will understand in our coming sessions. Okay, so now the question is most abundant genetic material. So most abundant genetic material. So here I have given you an idea. Okay, so what could be the most abundant genetic material? So you find it out and tell me the answer, right? So the most abundant genetic material. So here I told you the viroids have genetic material RNA. Viruses will have either RNA or DNA, but all the living organisms, they are going to have DNA as the genetic material, and RNA will do some other functions. So those functions I have mentioned here, right? Now let's see, this RNA and DNA are what we call them the nucleic acids. Call them nucleic acids. So the nucleic acids are RNA and DNA. So this, the full form of RNA, it will be ribonucleic acid, and another one called deoxyribonucleic acid. Right. So we have two types of nucleic acids, and first we'll study about DNA. Then later we'll discuss about RNA. So here let's look at DNA, the deoxyribonucleic acid. So here DNA is a long polymer of deoxyribonucleotides. So it is a long polymer of deoxyribonucleotides. Don't worry, I'm going to discuss in detail what actually are these ribonucleotides, I mean deoxyribonucleotides, and its structure and everything we will see in detail. So DNA is a long polymer of deoxyribonucleotides, and the length of DNA. So the length of DNA, it totally depends on the number of nucleotides present in that. So the length of DNA is, okay, will be equal to number of nucleotides present, right? As then, okay, more and more nucleotides add on, the length of the DNA will go on increasing. So if the nucleotides number decreases, then the length of the DNA molecule also decreases. So that's what I mentioned, the length of DNA will be equaling the total number of nucleotides present in it. And we can take here a pair of nucleotides is what I call a base pair. So one pair of nucleotides is one base pair, right? So later we'll see in detail about these nucleotides. Okay, so just at present keep in mind that if the number of nucleotides increase, the length of the DNA also increases. So let's see some examples in this case. Okay, so there is a pi x174. Okay, so it's called pi x174. So this is not an into, and okay, so no need to multiply the pi value with 174. This is a bacteriophage. Is a bacteriophage. Okay, so here we know bacteriophage is a virus that infects, right, bacteria. So pi x174 is its name, and this one has, okay, 5386 nucleotides. So that is 5386 nucleotides are present, okay, in this bacteriophage. So this actually makes us to understand that this is a one that has single-stranded RNA. So it has single-stranded DNA, not RNA. It has a single-stranded DNA, right? So SS, that stands for single stranded. So later we'll understand what is a double strand and single strand. All those things will get clear as we proceed, okay, into the chapter. Okay, so here pi x174 bacteriophage has 5386 nucleotides, and this indicates that this bacteriophage has single-stranded DNA. So it has single-stranded DNA. Then coming to another one called bacteriophage lambda. So and another bacteriophage, okay, lambda. This one has, okay, 48502 BP. So it has 48,502 base pairs, right? So here small b, small p, that stands for base pairs. So when I use that, it indicates that it has a double-stranded, okay, DNA molecule, right? So it has dsDNA. So that's what I'll mention here, ds, which means double-stranded. Okay, so bacteriophage lambda has 48,502 base pairs. Then coming to another one, E. coli. So the Escherichia coli, in this case it has, okay, 4.6 into 10 to the power of 6 base pairs. So you can see here, as I go on from the pi x174 to E. coli, the number of nucleotides are increasing. So it means that, so out of the three which I have mentioned, the one organism that has longer DNA is in this E. coli. So out of these, okay, three, and the smallest one is in this case pi x174. So we will include even humans also in case of humans. Okay, what will be the number of base pairs? So in this case we will see 3.3 into 10 to the power of, okay, 9 base pairs. So this is only, we have taken haploid content, not diploid content. If it is the case, then it will become 6.6 into 10 to the power of 9 base pairs, right? So when you see all the four, you can see it's the humans who have, okay, much longer DNA molecule among these four, right? I hope it's clear. And here Note point. So DNA is an acidic substance, right? So it's an acidic substance and is present in nucleus, right? And DNA as an acidic substance and present in nucleus is actually first identified by, so this first identified by a scientist named Friedrich Mischer. So he's the first person to identify DNA as an acidic substance, right, and is present in the nucleus, and of course in 1869. So he's the first person to identify DNA as an acidic substance and present in the nucleus. And at the time he and called it as nuclein. Okay, so he called it nuclein, which means the one which is present in the nucleus, right? So now we will understand the structure of DNA. So a DNA molecule is composed of, so a DNA molecule is composed of, right, nitrogen bases. So composed of nitrogen bases, then Pantos sugars, and then phosphates, right? So any DNA molecule is composed of okay, nitrogen bases, okay, Pantos sugars, and phosphates. I'll just mention, say, phosphoric acid. So of course that later will be forming into phosphates. How is all that? I will be telling you detail, right? So a DNA molecule is composed of nitrogen bases, pentose sugars, and phosphoric acids, right? So first we'll see about nitrogen bases. So here, as the name indicates, they are the one who will be having nitrogen. And here, these nitrogen bases are classified into two basing on the number of rings they have. So if they have single ring, so they are called pyrimidines. And if they have two rings, so double ring, and in that case they are called purines. So if the nitrogen bases have single ring, then they are called pyrimidines, and if they have double rings, they are called purines. So here I'll just give an idea so how the single ring will be seen. So this is the single ring, and in case of double ring, okay, so this is how we see in case of a double ring, right? So single ring, if they have single ring, they are pyrimidines. If they have double ring, they are called purines. So now let us see the pyrimidines here. So it includes cytosine, then thymine, and then uracil. Okay, so these are three types of pyrimidines. Of course, here you have to keep in mind the uracil is present only in RNA. So uracil is present only in RNA, right? So they are not present. That is uracil, which is a pyrimidine, is not present in DNA. Okay. So here the cytosine will abbreviate it as, okay, capital C, this as capital T, thymine, and this as capital U, uracil. I hope you remember when I told in the previous session, so how is a gene, so how does a gene look like. I told you it will be just a sequence of nitrogen bases, so where I have used, okay, this C and T. Hope you remember, right? Now coming to the double ring, okay, purines. So in this case we have, okay, two types. One is adenine, so we'll take it as capital A, and another one is guanine, so that will be capital G. So adenine and guanine. So here we have, of course, adenine and guanine that will be present in both DNA and RNA, right? So adenine and guanine present in both DNA and RNA. And coming to this cytosine, so this is also present in both DNA and RNA. Coming to thymine, is present only in DNA. So thymine is present only in DNA. So, okay, in case of RNA, the pyrimidine present will be the uracil here. Okay, so here we have three types of pyrimidines, in which, okay, only two of them will be seen in case of DNA. So that's what I'll mention. Nitrogen bases found in DNA. So those are one, adenine, then guanine, then cytosine, and thymine. So these are the four nitrogen bases that we see in case of DNA molecule. Next, so here a purine always pairs with the pyrimidine. So a purine always pairs with pyrimidine or vice versa. So you can say pyrimidine pairing with the purine or purine pairing with the pyrimidine. Okay, so here let's see adenine, that is adenine always pairs with the thymine. So adenine always pairs with thymine. So you can see adenine is, okay, purine, and thymine is pyrimidine. So this is one point. So here, so always this is to keep in mind. And next, we'll see about guanine. So guanine always pairs with the cytosine. Guanine always pairs with cytosine. So this is okay. So adenine, so I'm talking about in case of DNA molecule, right? Adenine will pair with the thymine, and guanine will pair with the, right, cytosine. So I'll mention that adenine always pairs with the thymine with the two hydrogen bonds. Okay, so between adenine and thymine, okay, so there are two hydrogen bonds. Or I'll mention adenine pairs with the thymine, okay, with the two hydrogen bonds between them. So adenine pairs with the thymine with the two hydrogen bonds between them. Then coming to, I told you guanine, it will pair with the cytosine. In this case, there will be three hydrogen bonds. So guanine pairs with the cytosine with three hydrogen bonds between them. Right. Hope it's clear. So here one more point, that thymine is modified form of uracil. So thymine is modified form of uracil, and for that reason the chemical name of thymine is what we call it 5-methyl uracil. It's called 5-methyl uracil. So here for understanding, I have drawn the molecular structures of uracil and thymine. So this is uracil and this is thymine. Okay, and when we see the numbering, so this is the first one, second, third, fourth, 5, 6. Okay, and same in this case, 1, 2, 3, 4, 5, 6. And you can see in case of thymine, at the 5th position there is addition of methyl group. So you can see here presence of methyl group. Okay, so you can see the remaining structure will be almost the same. So here the uracil, that's what I told you, okay, modified and formed, okay, thymine. So I say thymine is modified form of uracil, right? So that's about the nitrogen bases. Next we'll see about Pantos sugars. Okay, so here the name itself giving us information, Pantos sugars, which mean they have 5 carbon. So all these are 5 carbon containing sugars. Okay, so here the Pantos sugars, we have two. One is ribose sugar and another one is deoxyribose sugar. Okay, so ribose sugar and deoxyribose sugar. Now here, so the formula of this ribose sugar is C5H10O5, and coming to deoxyribose, it will be C5H10O4. I think with this you might have understood that why I have used, okay, the word deoxy, okay, in front of ribose, because there is one oxygen less. So you can see here, in case of ribose it is O5, and in case of this it is O4. So that is the reason why I call it deoxy. So oxygen less, okay, ribose sugar molecule. Okay, so let's see the structure of ribose and deoxyribose. And before that, one more point, this is present in RNA molecules, whereas deoxyribose sugar is present in DNA, is present in DNA. So first let's see about this ribose sugar. Okay, right, and so here OH, H, right, and here H, and in this case also it will be, okay, OH, of course, and just mention H, right, and in this case it will be H, and here it will be CH2OH. You have to keep in that here is one carbon, okay, so this is another carbon, here another carbon, and here another. So you see in this case, this will be the first carbon, this is the second carbon, the third carbon, this is the fourth carbon, and here is the fifth carbon. Okay, so here you have, I'm sorry, I forgot to keep O here. So this will be in case of ribose, in case of ribose sugar molecule. Now coming to deoxyribose. So here you will have same OH, H, okay, here H and H, and here OH, H, H, and CH2OH. So you can see everything is same, but except one difference, that here at the second carbon there is no oxygen, right? So here to keep in mind, the black dot indicates carbon. So here carbon, and so this is the first carbon, second carbon, third carbon, fourth, and this is the fifth carbon. So this is seen in case of deoxyribose sugar. In case of deoxyribose sugar, so you can see the same which I have mentioned, you will find in this case C5H10O5, and in this case you will find C5H10O4. So there is, okay, one oxygen less at the second carbon, and that is the reason why I am using deoxyribose. Okay, so the actual is ribose, but in DNA molecule, okay, one oxygen is less, for that reason I'm calling it deoxyribose. So why the DNA has one oxygen, the question comes, that we will try to answer in the coming sessions, right? So that's about the Pantos sugar. And the next one, the next component of DNA is phosphoric acid. So let's see, phosphoric acid. So this phosphoric acid, whose molecular formula is H3PO4, PO4. Okay, and the ionization will result in the formation of phosphates, that will be PO4 3 minus. So it will be PO4 3 minus. Now if you look at the structure of the phosphoric acid, so you will see, okay, double bond O, and then here you have OH, and here we have OH, right? And this becomes the phosphate, so P double bond O. Here the H, all these will be removed, so it will get a negative, so O minus, so here O minus, and here also same. So this is the phosphate. Okay, and of course due to presence of these phosphates, the DNA gets its negative charge. So due to presence of phosphates, DNA gets negative charge. So I say that DNA molecules are negatively charged. So that I'll just mention it. DNA molecules are negatively charged. So the reason why they are negatively charged is due to presence of phosphates. You can see with the negative charge on them, right? So that's all about the three components of DNA. Now the combination of these components will result in formation of nucleosides and nucleotides. So let's see about nucleosides and nucleotides, right? So remember in the beginning of this chapter I told you that the length of the DNA molecule will depend on the number of nucleotides present in it. Now let's understand what actually are these nucleosides and nucleotides. So here a nucleoside is a combination of nitrogen base plus Pantos sugar. Okay, so a nucleoside is a combination of nitrogen base plus Pantos sugar. Okay, and coming to nucleotide, so when coming to the nucleotide, this will be a combination of nitrogen base plus Pantos sugar plus phosphate. So you can see in case of nucleoside, you don't find the presence of phosphate. Okay, right? In other words, I can say nucleoside plus phosphate is a nucleotide. Okay, so I hope this is clear. Okay, so here a nucleoside is a combination of nitrogen base plus Pantos sugar, and in nucleotide there will be addition of phosphate. So if you are going to add phosphate to the nucleoside, then it will become a nucleotide. So how the nucleotide looks like? So here we know this is the Pantos sugar and this is a nitrogen base, and here we have the phosphate present. So here we will be having O, P, right, O, and right. So you can see here the presence of this phosphate, and here in this case I'm going to mention with the red color the carbons. So this will be the first, second, third, fourth and fifth carbon. So this is Pantos sugar, so which I'll mention with the S, it's a sugar, and this is nitrogen base, say suppose 89. Okay, we know 89 is a purine and we'll have 2 rings, and this is a phosphate. Okay, so you can see here the nitrogen base, right, a pentose sugar and a phosphate. You can take any nitrogen base here, so I have taken 89. You can take cytosine, you can take thymine, you can take guanine also, no problem. So whatever that nitrogen base plus pentose sugar plus phosphate, so all together will form a nucleotide. Right, so now let's look at the names of different nucleosides. So first we'll take here the nitrogen base 89 plus, okay, the pentose sugar in case of DNA is called deoxyribose. So 89 plus deoxyribose will result in a nucleoside called deoxyadenosine. Will form deoxyadenosine. Then next, guanine plus deoxyribose. Then it results in formation of deoxyguanosine. Will form the deoxyguanosine. Then coming to thymine plus deoxyribose, this will give, okay, deoxythymidine. Then lastly, cytosine plus deoxyribose, then it will result in the formation of nucleoside called deoxycytidine. Right, so for you a work, you need to find out the nucleosides in case of RNA molecule. Okay, whether it is RNA or DNA, a nucleoside is a nucleoside and nucleotide is a nucleotide. So in case of RNA, a nucleoside means it's a combination of nitrogen base plus pentose, and nucleotide means again for this we need to add phosphate. So find out, so as I have mentioned here names, so what names will come up in case of DNA, so if you are considering these nucleosides. And with this I will end today's session. In the next session we'll see about nucleotides and further explanation on DNA.